Hello and welcome everybody to my presentation for my master's thesis project on morphological analysis of histamine-induced human-derived serotonergic neurons. Let's get started. So we know that depression affects around 300 million people globally and its prevalence is steadily increasing. However, its pathogenesis is particularly complex, which has led to the appearance of a number of different hypotheses, which include the monoamine and the inflammation hypothesis amongst others, right? Recent research, however, suggests that histamine may play a role in synergizing both inflammatory and monoamine hypotheses. Now, serotonergic neurons have remarkable structural plasticity capabilities, being able to regrow and alter upon behavioral and neurotransmission alterations, such as during depression. And just from the literature, I wanted to highlight two key findings and the overall trends, which is one, the changes in innervation are observed in stress-induced rodent models, and these changes in innervation are observed throughout monoamine neurotic structure studies. And two, SSRI responses in patients are directly related with an increase in innervation, which means a better response, more innervation. Now, with histamine implications in both of those key points, the full picture as to whether histamine may play a part in this remains to be fully understood. In order to better analyze the changes that neurons suffer upon a specific kind of treatment, neuromorphology analysis techniques such as Scholl analysis allows you to do that. This is done primarily through the measure of neuritic intersections as the radius typically from the soma increases. And how Scholl analysis is currently done is mainly using semi-automatic approaches that aid tracing of the neurons, or using Autoscholl, which is a technique that automatizes Scholl analysis from segmented neurons. However, there is currently no fully automated open source high throughput method for analyzing the morphology of iPSC derived neurons. So all in all, this leads to our main hypothesis, which the main key point of this study, which we hypothesize that histamine exposure leads to significant changes in neuron structure, affecting their complexity and branching, which can then be correlated with innervation changes. In order to test the hypothesis, we will have three main objectives and aims. So first, we will perform immunostaining and imaging of both histamine-treated and control iPSC-derived serotonergic neurons. Secondly, we will develop a Scholl analysis software that is customized to analyze the morphological characteristics of these neurons. And finally, we will apply this tool to analyze the dataset and compare its performance with a manual analysis baseline. So firstly, our human iPSC serotonergic neurons are treated with 500 nanomoles of histamine for 3 days. We stain for DAPI and antibodies against neuronal markers such as 5-HT, MAP2, and tau, and tubulin 3. And finally, we collected with laser scanning confocal microscopy about 45 images of our PAMB1 cell line. So we had an external observer that was blinded to experimental conditions and performed tracings, particularly on 5-HT signal using simple MIRI tracer, using the DAPI channel as the root node for each tracing. So moving on to the automated segmentation software that we created, we have to understand one thing, that the starting point for Scholl analysis involves the identification of a central point of analysis, typically the soma, and tracing of the neuroids or a skeleton of the neuroids. So the process is outlined in the following flowchart. So what we do is we prepare the data for analysis, we identify 5-HT somas, and then using that, we segment and skeletonize those somas. Posteriorly, we do Scholl analysis and output the profiles and metrics. So just for the sake of time, we would just be talking about these key points, key blocks in the middle. So firstly, we need a way to segment serotonergic somas. So we follow three main steps. First, we apply auto-thresholding to the 5-HT channel to segment the strong 5-HT somatic signals, followed by the removal of small objects to further refine this segmentation. Secondly, we use watershed segmentation on the distance transform of the DAPI thresholded channel to effectively separate continuous overlapping somas. And thirdly, we look at the colocalization of the signals and keep only those somas that actually belong to serotonergic neurons. We also store their center of mass coordinates for posterior processes. Now, in order to pre-process and segment serotonergic neurons, we follow three simple steps. Firstly, we normalize and perform medium filtering on the FibreHD and MAP2 channels and perform a weighted combination to reduce FibreHD noise and enhance those colocalization neuritic structures, those colocalized neuritic structures. Now, then we use also thresholding and morphological operations to close the neuritic gaps and remove noise. Then we combine this result with a MAP2 thresholded image and enhance the structures using Sobel edges. Finally, using the previous identified serotonergic somas that we saw in the slide before, we perform region growing to gather connected masks of all the serotonergic neurons. So to obtain a robust topological skeleton of the neurons, specifically for irregular somas, we use the average outward flux algorithm. So the distance map, which is computed from binary images, we use that to compute the flux and richness measures, which are then combined to further refine the output skeleton. So going into the results, we find that immunostaining images show discriminatory FibreHD staining with regards to the dense neurons indicated by the MAP2 staining. Our software from a collected multi-channel image from obtained from confocal microscopy is able to identify serotonergic neurons, compute individual skeletons along with the centers of analysis, which are the somas, and perform shoal analysis and output the shoal profiles, quantifying the complexity of each neuron. If we look into the merged shoal profile results for all the traced or segmented neurons, first, I just want to clarify the x-axis of the plot shows the radius and the y-axis shows the intersections. So if we compare the manual analysis versus the automatic analysis, well, we see that in both plots we identify that there's a key difference between the control and histamine treated cultures, specifically an increased amount of intersections of the control group with respect to the histamine group. So we can see highlighted here. Afterwards, we find, well, interestingly, we do find an increase in distal intersections for the manual analysis, and the automatic analysis fails to identify this trend. And all in all, using linear mixed effect models, we find the same results for both approaches. The control group shows more intersections compared to the histamine group. To investigate where the automatic approach fails, we can look at this specific case. As we can see, our automatic approach performs comparably to the manual approach, specifically close to the soma. As distance increases, it starts underperforming, failing to identify long neuritic intersections. And in contrast, autoshoal, skeletonization, and center of analysis estimation fails to perform well compared with both the automatic approach and manual approach. So just before we discuss the final results, it's important to mention that due to the iPSC coaches being at an early developmental stage, we find significant colocalization of our MAP2 and tau stainings, which makes us unable to to properly distinguish between dendrites and axons. So therefore, we just talk about neuritic changes going forward. So our approach, all in all, manages to perform independent batch shoal analysis from user training, significantly reducing the user bias and improving over autoshoal, which still requires the user to segment the neurons. Now, our approach efficiently, and this is probably the key advantage, is that it efficiently analyzes the data, analyzing 45 images in about 3 minutes, significantly reducing the user time consumption and avoiding user tracing. Now, the key disadvantage that our approach has is that it lies in segmenting log neuritic structures with unsupervised techniques. Now, it does not implement any tracing techniques, and this is probably the primary source of bias of our approach with respect to the manual tracings. Despite having limited samples, I want to highlight the key findings we identified. First, we see that reduction of proximal neuritic branching while promotion of distal neuritic branching in histamine-treated neurons versus the controls. We also, from a statistical analysis, we show that a reduced number of intersections is found in histamine versus the control cultures, which can be linked to findings in the literature, particularly in stress-induced depression models. Now, all in all, we were able to successfully develop an approach that automatically quantifies serotonergic neurons complexity. We found evidence that points towards histamine actually playing a part in the reduction of proximal branching while simultaneously promoting neuritic growth. However, it is important to mention that to further improve on its performance, on the software performance, we should implement instance segmentation algorithms that can further improve the software accuracy.